So good evening, everyone. So first, I'd like to thank BioNano for the invitation and also for giving the opportunity to present this work on familial adult myoclonic epilepsy type 3. So I'm Christel Depienne. I work at the Institute of Human Genetics in Essen, Germany. And I'm really delighted to share this presentation with Mark Kopek, who has also been a very nice collaborator for us. So you just heard Mark presenting a nice overview on repeat expansions and repeat expansions causing fame, but I just want to remind you some important characteristics of these expansions. So familial adult myoclonic epilepsy is a dominant disorder that is characterized by an adult onset myoclonus associated with epilepsy in at least half of the cases. And it is associated with the same repeat expansions in at least 6 different genes that you can see on these slides. And these expansions are composed of 2 different motifs, a TTTCA motif, which is here in yellow, and which is actually the reference repeat present in normal individuals, repeated between 10 to 20 times. And the expansions also contain a repeat insertion of a different motif, 3TCA, which is depicted here in red. And this motif is probably the pathogenic part of the expansion. And here I'd like to mention that in some healthy individuals, some of the TTTCA is expanded and that it's non-pathogenic. So it's probably the TTTCA that is associated with disease. And so all these expansions are intronic, and all genes are expressed in the brain. But if you look at the encoded products of the genes, they all have different functions and cellular localizations and have really nothing in common. And so it's pretty clear that these expansions are probably pathogenic independently of the gene where they occur. And the gene itself is just a way to express the expansion. So I will talk about FAME3, so expansion in the gene called MARCH6 or March F6 because it has been renamed recently. And the story is very similar to Mark described for FAME2. We first mapped the locus in this very large range family more than a decade ago. And then there was a single locus on chromosome 5, encompassing 9 megabase and about 50 genes that we sequenced many times over the 10 years, and we couldn't find what was the causative variant until the FAME1 repeat expansion was found. So rapidly after, we could identify the expansion in MARCH6, confirmed it using repeat prime PCR assays, as you can see on here, and find additional families where this repeat expansion occurred and was associated with FAME. And of course, the first thing we wanted to learn about these repeat expansions is how large they were and what was also their structure. And then we used 2 different techniques. The first one is nanopore sequencing, and I will present the results. So if you're interested in those results, I would refer you to our paper published last year. And I will take a few minutes to present another method that we use to characterize these expansions, which is molecular combing, also known as fiber fish. And this technique is actually quite similar. So it has a common characteristic with BioNano optical mapping. So the first one is that you cannot use the DNA that has been stored in your freezer for many years. You have to have fresh blood samples or living cells to you perform new extractions. And what you do is to extract very long fibers and you comb them on cover slips, and then you simply do fish fluorescent in situ hybridization, and we very simply labeled the region where the expansion occurs in blue and green, so the flanking regions, and we also labeled TTTCA part, pathogenic part of the expansion in red. And this is the result in healthy individuals who you see always the blue and green stainings, and you never see red. And this is what you see in affected individuals with the expansions. So you see the same flanking regions with this red region in between, and you can appreciate the variability between different individuals which are here, and it's a quantitative technique, so we could use it to really quantify each part of the expansion. And looking at this individual who had the largest expansion with the problem of one of the family, we could actually see an amazing variability of the repeat expansions that you can see on these pictures. So some repeat expansions have no TTCA, some are interrupted, some are very large, and there is really an amazing variability that you can see also here under measurements. We also described actually some abnormalities that we call micro-rearrangements, where the red part was not between the blue and the green, or signals that had overlays. And we used these measurements to do the correlations between the age at seizures or tremor onset, and to show that actually it was the TTTCA part that correlated with the onset of epilepsy in our family. So what we did is, so just to come back at this slide, so this is a very nice technique, but the drawback of this technique is actually that it's a lot of work to develop it just for one assay, one regions, and we have at least 6 different to study in FAME. And so, of course, we were interested in bio-nano optical mapping because of its ability to detect all FAME repeat expansions at the same time, the genome-wide level. So first, we wanted to be sure that the technique is able to detect those expansions. So we took the same agarose plugs that we use for molecular combing, and of individuals of who had different sizes of expansion. And so we sent the samples to San Diego, and they did the experiments. And so they extracted the DNA from agarose plugs. They labeled it with the DILI-1 enzyme, and they performed an assembly and also a de novo assembly and a rare variant analysis pipeline. And of course, the questions were, is optical mapping able to detect the repeat expansions that we saw in these patients, and do the results match the previous findings that we obtained with molecular combing? So for those who don't know, who've never seen a result from bio-nano, just to mention that actually this is a very powerful technique that is able to identify really many CNVs, and so you should be prepared, to filter structural variants. And you see, if you don't filter all the structural variants, that you can see more than 4,000 variants of different types. So what we use is actually the control individuals that bio-nano offer. So you can use 200 control individuals to filter your data. And if you do this, and also you keep only the structural variants overlapping genes, then you find a number of structural variants that you have to look at, which is really decreased, or let's say about 30 CNVs or structural variants altogether. And here it is, the insertion that corresponds to the repeat expansion, and it's actually the only structural variance that is detected in our locus, and also it would be the only structural variant detected in more than one individual. And this is how the expansion is seen by optical mapping. So it's actually the same individual for which you have seen data previously, the one with the very large expansion. So using bio-nano, we could detect an expansion of 40 kb, which was exactly the same size that was detected by molecular combing. And you can see when the molecules are sorted according to their size that actually they are all different. So the somatic mosaicism is really really huge, as we previously we saw with molecular combing. And I want to just highlight here that there are several molecules that are broken just at the site of this expansion, and we think that it's the repeat expansion actually constitutes a fragile site. So I said that we also use the rare variant analysis pipeline. This is what you see on this part of the slides. And as you can see, this pipeline is also able to detect the somatic mosaicism, although the molecules are beamed into arbitrary categories, as you can see here. So what about the other samples? So the good news is that we detected the expansion in all samples. So for those samples, we had a bit of variability of the mean size of expansion compared to what we detected or what we calculated with molecular combing. So here with BioNano, we had 5 kb in this individual for a size of 7 kb previously calculated. We could see also the somatic mosaicism in this individual, and this 13 kb using BioNano, 6 using molecular combing, and we also detect the somatic mosaicism. And then in this individual who had the smallest repeat expansion, calculated around 5 kb, it was actually 2.7 kb detected by BioNano, and no mosaicism was detected because the rare variant analysis pipeline detects mosaicism for structural variants above 5 kb. So my conclusion, optical mapping is a very efficient technique that can detect the same repeat expansions at a genome-wide scale, which is extremely useful for us because we don't have to develop different assays to diagnose these repeat expansions. We could confirm the somatic mosaicism using both the rare variant analysis pipeline and also just looking at the molecules themselves. If you want to do gene discovery with BioNano, you really need control individuals to filter the variants or to just analyze multiple samples or have an idea what you are looking for, because it's you detect a lot of structural variants with the techniques. And the drawback of BioNano is, of course, that you cannot detect what is inserted. And in the case of FAME, it's important because we really need to know that there is a TTTCA repeat expansion part that is the pathogenic one. So we need to combine optical mapping to different technology, a sequencing technology, to be really able to say that there is a TTTCA repeat expansion at this site. And finally, I just want to mention that the resolution of the technique is around 500 base pairs, which is sufficient to detect the large repeat expansions, but which is actually insufficient to detect the small repeat expansion, including all or most of the polyQ, polyglutamine, polyalanine expansions. And this is an overview of the repeat expansion disorders. And just to highlight the ones that could be detected by the technique, it's the ones that are in green. So these ones would be for sure detected because they are above the threshold for detection. The orange ones are the ones that could be detected if they are large enough, but some of them could be missed because they would be too small, and the red ones would not be detected. And in the case of FAME and also Canvas, which is another expansion, repeat expansion disorder, there is the problem that the pathogenic motif is actually different from the reference motif, and some individuals have non-pathogenic expansions. So of course, it's important to make the difference. And I will finish my talk by thanking all the people who were involved in this study over so many years. So people from my group, Raël Florian, Sabine Kaya, Cécile Saint-Martin, Caroline Nava, Boris Karen, now Christopher Schroeder, and the FAME consortium, including Mark and Joseph, Melanie, Ingrid, and so many others that are not even on these slides. Big thanks to family and to funding agency, and also to Bionano for helping us with this resource. Thank you, and happy to answer your questions.